Hey biology students, we are working on deer mouse fur color, activity 1, lesson 1.1. We talked about the sampling of taking mice from their burrows and some soil from inside the burrow and at the entrance and measuring the value of soil brightness. Brighter, whiter soils, lighter soils have higher numbers and darker soils have lower numbers. So we ended up talking about some of the data that was collected for this study, and you were supposed to list all of the variables and then pick two that were related, that you thought might be related. For example, as if one variable increased, the other decreased, or if one variable increased, the other variable increased as well. They had some sort of predictable relationship between them. So now we get to look at and analyze the data a little bit further. We have a map of all the different places where mice were collected. And we have scatter plots made from some of the variables in the table. And a scatter plot is just like it sounds. You just graph the data, and sometimes it looks very scattered. Sometimes the data looks just scattered all over the place. And sometimes it looks like it's a little bit more like someone was practicing darts and they wanted to have some kind of pattern here. This looks like maybe my dart playing where it's just all over the place and doesn't have any sort of relationship. It's just plain random. These graphs look like there might be a somewhat purposeful organization of those points, which represents a relationship, a predictable relationship between the two variables. So if you knew one variable, like you knew fur brightness, could you predict with some degree of certainty what the surface soil brightness would be? Or if you knew the subsurface soil brightness, that's the soil taken from within the mouse burrows, would you know, or could you predict with reasonable certainty the surface soil brightness, the soil taken outside the burrows? So question 7 says, do any of the graphs show meaningful relationships? So a non-random pattern of dots that might even be sort of linear if you use your imagination. Linear means like a line. And then question 8 means, based on the data and graphs, what can you claim about mouse fur color? So here's fur brightness, here's fur brightness, here's fur brightness, here's fur brightness, here's fur brightness. Which ones have patterns that are somewhat predictable, maybe sort of make a line? If you know one, you might be able to predict the other. So that's what you want to think about for question 8. If you know the fur brightness, can you predict with some degree of certainty what the subsurface soil brightness is going to be? What questions from the driving question board are you interested in exploring next? Some of the things we talked about in class are how do predators influence mouse behavior? And what are the chances that a mouse with fur that does not match the environment, what are the chances they're going to survive? We talked about the high visibility workwear that some people wear on job sites to keep them safer. Some of you may have seen folks wearing t-shirts like this that are working construction, or maybe they're working transportation job, or they get out of their truck and they put on a bright yellow vest so people can see them. we talked about that would not be good if you were a mouse. Because as we're going to see in the next part here, lesson 1.2, there are predators around that might snap you up. If you're a mouse that's this color, you might be someone's lunch. Here are some of the other questions that students ask that you might be interested in exploring further as we continue in this lab. Pause the video and check out all the different questions. All right, now that you've finished question 9, we're going to head over to activity 1.2. You can download your answers by clicking Show All Answers and then following the directions in the lab sheet that's in Schoology for the lab. So there's a cool simulation here that we get to kind of play around with a bit. We're going to make observations with the multi-level simulation. And you'll see why it's called the multi-level simulator a little bit later. You can look at the population level, you can look at the cellular level, so we can look at genetics. There's a lot of different aspects of a population you can check out with this particular model. But what we're going to do right now is just kind of play with the model. Let's look at the first question, and then we'll look at in more depth at the first model here. Our job is to just kind of experiment with the model. Click Run. Maybe we want to add some hawks. And you guys can see what's happening here. Other things to observe while you're watching. How much time is passing? What's happening to the mice over time? How long does a mouse live? What happens when you add hawks or take them away? Does the environment make a difference? You can change the environment here. You can add the hawks back in and see what happens. So when we're running the model, we can also turn on the graph here. There's also a pie graph if you prefer that. And you can see that we started at 1/3 light mice, 1/3 medium mice, and 1/3 darker mice. And after 95 months, we have a much different distribution here. My advice would be keep just one environment and let it run, and then reset and run again in a different environment. We can also we can inspect. So if you click on a particular mouse, it'll tell you the genotype, it'll tell you the sex, it'll tell you that particular color. So you can click on a bunch of mice and learn about them. If you want to collect, you can grab a mouse and take it over here. And this is a good way to sample your population. If you want to kind of check out what's going on with some of your population members here. There's color codes that say female. Those are the yellow circles, and male. Those are the red circles. If you want to go back to the graph, you can just hit run again, or you can pause. So now you know basically how to use the simulator, and that's going to be important for the next several days of lab. And we'll learn more about the simulator as we keep going. Let's head to the next page here. So it's time to collect and analyze some data to help determine what influences fur color in deer mice. Read the instructions in the simulation below to set up and run experiments. In your Deer Mouse Field Notebook, fill in Lesson 1.2, Table 1, as you experiment and collect data on the number of mice with different fur colors over time. The table can be downloaded in Schoology. It looks like this. You can fill it out in Word. You can fill it out as a PDF in Notability. But do turn it in with the rest of your lab. So now that you know what you're looking for, you're going to run 3 trials in the field. That's the dark background. And you're going to run 3 trials on the beach. Each of those trials, each of those experiments are going to have hawks in them, and they are going to last for 60 months. We'll learn a little bit more about that in a moment. So in order to fill out the table, we have to use, well, I'm going to reset here. We're going to make sure that we add hawks. We're going to use the beach 3 times, and we're going to use the field 3 times. To get exact values at particular points, place your cursor over the dark point on the graph. Take the average of each set of trials, then answer the questions in the left side. So let's get this going. Let's put a graph on. Let's add some hawks. We're going to start with the field because that's in the table first. And we're going to run this for 60 months. So we'll watch the months go by here, and then we'll look at our data. Notice how I added the hawks right away. And I want to do that. I want to add the hawks at the same time for every trial. Don't wait 12 months and add hawks, because then your trials won't be the same. So what's happening to our number of mice here? Sometimes it's clearer if we look at the pie graphs. We started at 1/3 of everything. And look at here. We're mostly at dark mice here. What do you think if you lived in this particular beach habitat? Would you want to be a dark mouse or a light mouse in terms of surviving hawk, being a hawk lunch? All right, now we can fill out our table. So we go to trial 1 of the table, and we fill out the number of light mice. Oh, let's go to 60 months here. So that's 60 in the beginning of that 2 coordinates. The 60 is the month, and the 43 is the number of mice. So here's our data table. We're working in the field environment. That's the darker environment. The beach is lighter. We just counted the dark mice, and there were 43. I like to make my answers a different color so my teacher can see them better. Let's go figure out how many medium brown mice there were and how many light mice there were. We need to go over to our graph. At 60 months, we click here, 60, that's 60 in the pair of numbers is the number of months, and the 4 is the number of light mice. So we can go back to our graph, or excuse me, light brown mice, or medium mice. So there are 4 medium brown mice. And then let's go back to our graph and see how many light brown mice there are. And I see that light brown right down at 0. And I don't even see the light brown color up here. So that helps reinforce the idea that there are no light brown mice. So I'm going to put 0 here. And I'm going to let you do the next 2 trials. It's okay if your numbers don't match mine perfectly. If you don't get the same numbers for trial 1, trial 2, and trial 3, it's okay. The sim is built that there's some degree of randomness in it, and you're going to get some different numbers here. Once you have your numbers, take the average. So to take the average number of light mice, I'm just going to put in some ridiculous numbers. Don't take these numbers because I don't want you to use them. So these are some ridiculous numbers. Don't copy them. I'm going to say that again. These are ridiculous numbers. Don't copy them. But I'll show you how to average. I'm going to take 0 plus 35 plus 400. That gives me a total of 435 if I add all of the trials together, and then I divide by 3. 435 divided by 3 is about 130 plus 7, about 137. I'll do it on a calculator just to make sure. So I'll put my calculation there. 435 divided by 3 is equal to, I was a little bit off, it's 145. Now that's not the right answer. It's not the right answer. Those are ridiculous numbers. I just put those in as an example. You should find numbers that are much lower than 35 or 400 when you do trial 2 and trial 3. But when you want to get an average, add the 3 numbers and divide by 3, and that'll give you the average number of light mice for your 3 trials in the field. Do the same with the medium mice and the dark mice, and then we'll work on percent. You need to do the same thing for the beach. I'll do the first trial for you. So to do a trial for the beach, I'm going to hit reset. I'm going to hit beach. I'm going to add hawks from the very beginning, and then I'm going to hit run. Now notice we start with 1/3 of each type of mice. That's not reality. That's not what nature would have, but we're doing this as a sim, and so we're trying to always start with the same and see what happens to the population over time. You can see the months are going by. I don't see any more dark mice anymore, and I don't even see any more medium mice. I see a lot of very light colored mice. That light colored line here. I'm going to hit pause here. I see at 60 months, I see 47 light colored mice. So now I can go to my table on the beach. I had 47 light colored mice. And I didn't see any medium colored mice, nor did I see any dark mice. Now you might not get the same results. You maybe will have some dark mice. Maybe you'll have some medium mice. I don't know. It just depends. And then do 2 more trials, reset, add your hawks right away, do it on the beach, do 2 more trials, take your average, and then we'll answer the questions that are part of the lab here. This is also part of the lab, but you'll hand it in a little bit separately from, you'll still hand it in the same assignment folder, it's just a different sheet. Question number 2 says, describe how the numbers of dark, medium, and light colored mice in the population change over a single trial. Now, will your answer be different for the beach versus the field? You'll have to figure that out. Compare the average percentages at the ends of the trials with the percentages from the beginning. How similar or different are they? The population always starts with approximately 33% of each color. Well, let's go look. This was our beach trial. We started with 33%, 33%, 33%, and we ended with 100% light mice. You can run another trial in the field to figure out what happens when you let the trial run in a field. What happens to these percentages when you're running the trial in a field. Next time, we'll go over how to do the average percentages if you don't know, and we'll go run a trial without hawks on the next page. See you later.